IB revision video for genetics, and this is part 1 of 2 because it's such a big area. This will be quite a long video, so obviously please split it up and treat it in individual sections. This is also not enough, definitely, to make sure you get the grades. It will be a supportive video, but there will not be enough time to go through all the different genetic crosses. You need to be familiar with lots of words before we start this, so you need to make sure you can identify what the different terms are. So if we start off with this term, an allele that controls the development of a characteristic when it is present only in one of the chromosomes. So that will be a dominant allele, so it is always present. Next term, a cell which the two alleles of the same gene are different, so you've got a cell with the genes and they have two different alleles, so that is heterozygous. Next one, a different form of a gene, so that is the term allele. Next one, a sequence of DNA that codes for one polypeptide, that is a gene. That can also code for a functional RNA. A change in the base sequence of DNA resulting in a new allele is a mutation. An allele that controls the development of characteristics only when the dominant allele is not present is obviously recessive. And a cell that contains one set of chromosomes so it's got a complete set of chromosomes, but only half effectively, that is your haploid. Go back to red, a genetic makeup of an individual is its genotype. The characteristics of an organism resulting from the alleles plus the environment is its phenotype. And a cell which with both alleles of a gene are the same is homozygous. And then a cell that contains two sets of chromosomes, as like humans, a full 46, is diploid. So you need to make sure you know all those terms. So what we're going to be looking at is genetics, obviously, and the basis of how genetics work. So to remind you, a gene is a sequence of DNA bases that codes for either a polypeptide or a functional RNA. Every 3 bases in DNA is called a codon. It gets converted in transcription to messenger RNA. Every 3 bases codes for one amino acid, which makes up the primary protein structure. So hence a different allele can result in different proteins, which results in different phenotypes. Alleles are a different form of a gene. So we eye color, for example, red eyes, brown eyes, or green eyes, and they can be dominant or. But don't forget, in all this, there are factors in play that are not just genes. The environment also has an impact on the phenotype, and that is therefore important. As I said, alleles are different forms caused by different sequences in the DNA. In some cases, 2 alleles can both contribute to the phenotype, and we'll talk about that later on. It is called codominance. You will notice that there is a genetic diagram convention, and that is the first thing you should always put is the parent's phenotype, followed by the parent's genotype, followed by what will be present in the gametes, followed by the genetic cross with the offspring genotypes, and then finally the offspring's phenotypes and race. So for example, here you can see this being played out. You can see that we have the parent's phenotype, parent's genotype, the different gametes they can produce, the genotype of the offspring, and obviously you might draw a genetic diagram here, and then the phenotype of the offspring, and in this case there's no ratio because it's 100% purple. If we took that example we just had, and all the purple were then what we call heterozygous, okay? The purple heterozygous would go together, so that again is the genotypes of the parents. We then show the gamete of the parents and draw a genetic cross like that. I wouldn't probably draw it like this myself, okay? And then we've got the genotypes of the offspring. So we can see that 3 of them would have the dominant purple allele here, dominant purple phenotype because they've got a capital A, and only one of them would have 2 recessives, giving the recessive phenotype. So there is a 3-to-1 ratio of dominant to recessive, as is normally the case for monohybrid crosses. Now, when we represent gene alleles, we need to represent them in a similar fashion each time. So if it's just 2 alleles for a gene, you always show the dominant and recessive with a suitable letter, and generally, obviously, the dominant would go first. If more than 2 alleles are in a population, a superscript is used. So for blood groups, you might have A, B, or O. So you can see here they've got I for all of them, and then A, B, and O. And the same is used in codominant. Let's have a look at some examples for monohybrid crosses then. So here we have Mendel's peas. Okay, so Mendel studied lots of different things, but pea pods was one, and they had green, which were dominant and found most common, so represent them with a capital G, and recessive yellow, which again we use the same letter, so you don't use a Y, you use a little g. So the dominant we assume have been purely bred, so we've got 2 dominants, 2 recessives. They then have the parent or offspring, the gametes of the parents, and then we can do a genetic cross to see how many of each offspring we get. So you can see here, all offspring would be green because they all become heterozygous. If we take that generation and we breed those 2 together, we end up with different gametes again, and then we end up with the following genetic cross. And this time, in the second filial generation, as always, monohybrid crosses, we have a 3-to-1 ratio. So we have 3 dominant phenotypes with capital G's and 1 recessive, which is your yellow pods. So you'll have a 3-to-1 ratio of green to yellow. And again, if you don't understand that, you need to go back and look more detail at it. And this follows what we call the law of segregation, which is in diploid organisms, characteristics are determined by alleles that occur in pairs. Only one of each allele can be present in a single gamete. This can explain different forms of conditions, such as cystic fibrosis, which is a mutation in a recessive allele on an autosome, and it causes an incorrect shape of a chloride transporter, preventing chlorine and water to be transported across membranes, and as a result, mucus builds up, and so do infections. You have lack of digestion, maybe infertility because of this condition. This can be shown in a genetic diagram as so. So you can see here 2 carriers of cystic fibrosis where they've got 1 dominant, 1 recessive. They do not have the condition themselves. But then when they have children, there is a 1 in 4 chance that a child could inherit both recessive alleles and therefore become a sufferer. So if we take that forward, we then need to look at the genetics of codominance. So we're still using monohybrid codominance. So monohybrid crosses involve dominant recessive. However, the alleles controlling ABO blood groups are different. So the ABO blood group inheritance basically means that red blood cells contain glycoproteins which determine their blood group. There are 2 forms of protein, the A and B antigen, and the gene coding for the protein is found on chromosome 9 in humans. 3 alleles actually code for A and B, or no. So here you can see we have A. Remember we said earlier on, if it's got more than 2, it's got to have this. So, or if it's codominant, it's got to be represented by like this, B, and then neither A and B. Okay, so this relates, as I said, to the polypeptides on the cell surface, which act as antigens. And if you have the wrong polypeptide, it will trigger an immune response, which is why blood groups are important in blood donation. So here you can see how it sort of works. This one we've got double A. So they will be group A. This one we've got AO, so that is group A as well. BB is group B, BO is group B, AB is group AB, and OO is group O. And that is because they'd have neither the A or the B antigen. So in this respect, A and B are codominant of each other, but both of those are dominant over the O. So the I represents what we call the locus, and it's a letter of, the other letter is the allele. So the locus is a position on the chromosome. A and B are dominant, as said. O is recessive. Sometimes gene has more than two forms. In this case, we're going to say they're multi-alleles. However, there are only ever two chromosomes in the homologous pair, so it will always it follows only two of the three or more alleles in existence will be present in a single organism. So that is the example we've. So why are the blood groups co-dominant? Well, all three alleles code for the production of the glycoproteins. So IA alters the glycoprotein by adding something called acetylgalactosamine. People without this allele make anti-A antibodies and would reject that. IB adds galactose. People without this will make anti-B ones, and people who have IA and B would neither make anti-A or anti-Bs. And O adds neither, which is why blood group O is such a useful one in blood donations, because it will allow you to add it for everyone. So here's some examples then. So someone, if we have looking at offspring and inheritance, so we've got someone with I and A. So again, we just treat this like before. You just take one A in a gamete, one B in a gamete, and then O and O here. So that one would be A and O, B and O, A and O, B and O. So you'd have 50% chance of each. And again, this works across all the different methods, just applying the same method as you have done before. Talk about this in cattle or horses and colour of coats. So here you can see roan cattle or horses. So this is roan here in the middle, where there's a mixture of two, they're not completely brown, but they're not completely white as well. And this is because the red hairs, so not brown, is R, and the W represents the white hairs. They are both dominant, so they end up being a mixture if you get both alleles of. The coat colour in horses is the same, so it's the same all different types of animals. So here you see a genetic cross between a red and a white cow and bull. So they've both got the two homozygous alleles. They're both, however, dominant, so all the offspring of the first generation will be CRCW, they'll all be roan. If you breed them two together, then you get this mixture, so CW, CW, so white, CR, CW, so roan, roan again, and red. So you get a 1 to 2 to 1 ratio of white to co-dominant roan to red. There is also something called incomplete dominance that can happen where there is an intermediate product formed. So here you can see we have red flowers, which are capital R, W, W for white, and they come together to make CRCW, and that is an example of incomplete dominance as an intermediate product has been formed. So you could get different examples such as this, so you could be asked to look at the different complete genetic diagrams. So remember the pink flower here is CWCR, so if you bred two lots of those together, you would have this combination here, so CRCW, CRCR, CWCW, CRCW, okay, and we're going to a 1 to 2 to 1 ratio here. And then remember the white flower has to be, so here we've got CRCW, and this one would have to be CWCW. So again, you just follow the genetic code and you would end up with a 1 to 1 ratio of pink to white flowers as a result. So the final section then we're going to look at is sex linkage. Now sex linkage is sex linkage, we need to understand how sex is determined. So really GCSE, so males in the sperm produce an X and Y chromosome, and females produce XX. So there's a 50% chance that you get either, because neither are dominant. So XY is always male, XX is female. And again, different way of showing the same thing there. So when we're talking about sex chromosomes, we need to know a little bit more about them. So the male is XY, okay, and one gene on the Y chromosome called the SRY gene causes the embryo gonads to become testes. This is called the testes determining factor, and the testes then start testosterone secretion, allowing other male organs to develop during puberty. So that explains why the Y chromosome leads to male. obviously no Y chromosome, so no testis determining factor. And the fallback position then, if you don't have this release of this hormone, is that the gonads become ovaries. Ovaries release estradiol, which causes female reproductive system to develop, and the female hormones start to be released at puberty. Sex linkage is when there is a genetic disorder normally associated with the X chromosome. So people often mistake this. Very few examples are there where it's linked to the Y chromosome because there are so few genes. So in your specification, we're only talking about X chromosome sex linkage. And basically, genes with loci on the sex chromosomes rather than the autosomes are the ones that cause sex linkage. We said a moment ago the Y is so small, it doesn't have many genes. The X is big, so it has much more genes. It's not homologous because there isn't another one in males. So the inherited pattern will depend on whether you're male or female. Genotypes that are recessive, therefore, are more common in males because you only have the X chromosome once. So you don't have, if you have a recessive copy, you don't have another version of it on the Y chromosome, and therefore it will show in your phenotype. So an example of a sex-linked disease is haemophilia A. Haemophilia A is when the blood clotting protein factor called factor 8 is coded for, and that gene is faulty and it causes haemophilia, meaning the blood takes a long time to clot. Bleeding it happens in joints, disabling internal bleeding will happen until recent medical advances. And these are the different phenotypes you could have had. So if you were genotypes you could have had. So if you were female, you could be normal. So the dominant H is healthy. Remember, because we're looking at sex linkage here, we've got to put it on the X or Y chromosome. You'd have a normal clotting factor here, but you've got a carrier, so you could potentially pass that off to an offspring. And if you're X little h, that would be lethal for females because when they start puberty, they would die due to the menstrual cycle. For males, you can either be dominant H or recessive H. So you can be normal clotting blood factor or you can be haemophilia. And obviously the majority of males are capital H, but you only need one recessive H because you only have one X chromosome to have haemophilia. So if we look now at what could happen then if we have a carrier female, so someone who doesn't potentially even know they've got it, and a healthy male. You can see the female will put both a capital H and a recessive H forward, the male only a capital. But because of the X and the H's, one of the offspring, if they are male, would be a sufferer. So you can see here there is a greater chance if you are male than female at having one recessive H. So here you would have a ratio of 1 to 1 to 1 to 1. And here, if you got it the other way around, if you had male who has haemophilia and a normal female, the male would pass on, definitely pass on one of those. So if you had a daughter as an offspring, obviously one of the X's has to come from the male, and therefore they would be a carrier female. If you were a male with haemophilia and you had a boy as offspring, you wouldn't be passing your X on. You would obviously only pass on the Y to the boy because the female passes the X on, so therefore the boy would be normal. In the case of a family tree. So often they use the royal families of Europe because for many years, due to levels of inbreeding, they were linked to high levels of haemophilia. So here you can see you'd be given a key. So a circle with a line for half of it through is a carrier female. A male haemophiliac is a letter here. So you can see here Victoria was a carrier. She had children with Albert, who was healthy, a normal male, I should say, without the condition, and they had a son who was a sufferer. So that son must have inherited as you would expect, the X from his mom, which had the recessive condition on it, and obviously the Y from the dad. The female must have inherited because obviously there's a healthy X and a little x, the X with the condition from the mom, and obviously the healthy X from the dad. And that's the idea. You might have to go through and work out what the chances of different people are. So for example here, Morris we don't know about, but you might be asked to calculate what was his chance of being a carrier or a sufferer. Different versions of the same tree, very common questions, so you need to practice examples of them. Linkage is also common in less serious diseases, so colour blindness is sex-linked. If you can't see the letter number 74 here, there is a chance you are colourblind, and effectively it is again caused by a recessive allele in the X chromosome. So here you can see we have a female who is a carrier. That female then passes on that little H to her son, and again you're more likely to be a sufferer if you are a boy. Another example of a disease which is caused by this is gene muscular dystrophy, and again it's on the X chromosome, coding for that dystrophin muscle protein. It's a large protein needed for muscle contractions. When you get the recessive allele, it leads to seriously truncated or non-dystrophin muscle. Muscles tend to degenerate, causing muscle weakness and eventually early death. And you can see here a different version how they've done it, but this is the abnormal chromosome, so again your recessive one. And that you can see again has the inheritance pattern. So if you had a son, if the mum would then passes that on, he would be a sufferer, whereas this son wouldn't. And then daughters, you could have a carrier. So the same genetics as before, just for a new. Also, from this then you would expect that females would have twice the number of X-linked chromosomes expressed compared to males, because you'd think that, well, if females have 2 copies of everything, that would mean that they would have twice as many chromosomes, because males don't have that. There does seem to be a mechanism that randomly makes the chromosomes become inactive. So one of them stops becoming active and they don't have this double impact as the rest of the autosomal chromosomes do.